and I will be talking to you today about neuroscience, the complexities of our brain and the invisible processes that go on beneath the surface. So when I was planning this talk, I realized I had no idea what my peers outside of my psych classes really knew about this topic that I care a lot about. So I asked around, and I figured out that you can make friends really, really quickly if you go to a party and you talk about science. So what you do is that you look around and you see who's having the most fun, and you go up to them and you say, hey, tell me what you know about neuroscience. So I did this last Friday, 11 p.m., really great time for an academic conversation, and I got some really interesting responses that helped me shape where I wanted this talk to go. Some of them being, I think of really smart people in lab coats, I think of chemists, I think of slices of the brain. And my favorite response, very simply put, was, neuroscience? Elyse, I don't know, I'm not a nerd. So, today I want to talk to you about why studying neuroscience doesn't have to be all about lab coats and chemicals. I want the entire BC community to care about neuroscience, particularly about the neuroscience of memory, because it affects us each day. It can help us perform better as an institution, and it can teach us valuable lessons on how to eventually avoid cognitive decline. And it doesn't take a nerd to realize that these topics are applicable to all of us in this room. So it almost goes without saying that as members of a higher education institution, we use our memories every day. We memorize information for exams, we meet new professors and peers each semester, we learn the locations of classrooms and campus dining halls. But we all know that cringeworthy moment when you walk into someone's spring semester that you know you met in the fall semester and you can't remember their name. And this leads us to my second point, being how can we ace our exams with ease, and how can we consistently remember the names of people that we met last semester? So cognitive neuroscience may hold these answers. There's a plethora of research that can teach us biologically backed techniques to form memories efficiently and prevent their decay. And that brings us to point number 3. As much as we don't like to think about it, we are all getting older, and it seems like neurodegenerative diseases such as Alzheimer's and dementia are unavoidable. But what if there was a way to keep your memories intact into old age? Before we can answer this question, we need to understand the basics of how the brain communicates. So I'm going to start with a few key terms. So today I'll be talking a lot about neurons. A neuron is a cell that is specialized to receive and transmit information in your nervous system. You may soon be able to see a cartoon version of a neuron on the right-hand side of the screen. There are about 100 billion of these cells in the human brain. There are branch-like structures extending from the cell body of the neuron in that top right corner. And the dendrites receive electrical signals from other neurons. These electrical signals have the ability to trigger the receiving neuron to send a message, also called an action potential, down the axon, that yellow strip that you see right there. This action potential is the mechanism that is ultimately responsible for transmitting neural information and for interneuronal communication. In other words, you could think of the action potential like fire, and the axon is a wick. And if the fire is hot and strong enough, it will continue burning down the axon and ignite the neuron that lies beneath it. So after hearing about this process for the first time, I was very confused because I didn't understand how we can make sense of 100 billion neurons talking in our brains at once. Luckily, one of the basic principles of brain organization, called localization of function, answered this question for me. Localization of function dictates that specific functions are served by specific areas of our brain. So I'll break down the human brain into 4 sections called lobes, and then show the location where memories are actually formed and housed. So here's a diagram of the left-hand side of the human brain. This person's head would be positioned like this, with their forehead facing the left side of the screen and their neck extending downwards. So as you can see, each lobe has a different function. The frontal lobe is in charge of many traits that make humans human. It's in charge of higher level cognition, such as reasoning, motor skills, and impulses. As you move further back along the skull, you find the parietal lobe that's in charge of tactile information, like pressure, touch, and pain. In the back, we have the occipital lobe, in charge of visual processing, such as colors and words, and the temporal lobe, in charge of auditory processing, like speech and music. So how do these lobes relate to neurons? Well, if you were to get a paper cut in class, which is a tactile stimulus, you would have increased neuronal activity in your parietal lobe. If you are presented with flashing lights and loud music at a concert, your occipital lobe and your temporal lobe neurons will have increased firing. However, you might also experience strong emotions when you get that paper cut or when you attend that loud concert. So let's dig deeper into the brain to find out where neurons fire to form these emotional memories. If we were to peel back the left side of the sample brain that I showed in the last slide, we would find the limbic system. The limbic system is a set of structures that lie deep beneath the brain surface. They bind together the information from your four lobes into a holistic experience. And it includes structures like the hypothalamus that governs physiological functions like hunger, the amygdala, the center for your emotions, and the hippocampus, which plays an essential role in your memory formation and recollection. As you form memories, neurons in your hippocampus, in particular, become active during 3 processes: encoding, storage, and retrieval. So let's go through the process of making a memory as if you are meeting a blind date for the first time. Encoding is the crucial first step to creating a new memory. This begins with perception through the senses, such as the sight of your date's face and the touch of their skin as you shake hands. This memorable and emotionally charged event causes your neurons to fire more frequently, perhaps in your amygdala, the seat of emotion, the frontal lobe, the seat of attention, or the thalamus, the seat of impulses. So this is where the hippocampus comes in. All of these inputs are combined into one single experience that we call a memory. Your brain will now decide how the memory will be stored, depending on its importance, which neurons it activated, and what kind of information it holds. If the state is particularly emotionally charged, you may store it for decades, and this storage enables you to recall it through memory retrieval. However, sometimes our memory storage fails us. We know we studied the date of a famous experiment for a final exam, but when the exam comes, we just can't remember. So why does this happen? We'll do a quick experiment so you can feel the reason for yourself. So I'm going to split up the room in half. On the left-hand side, I'm going to ask you questions about one event in your life, and I'll ask the right-hand side a different question. So I'll start with the left-hand side. I would like you all to silently recall the last time you went to the doctor for an annual checkup. I would like you to raise your hand if you can remember any of the meals that you had the day you went to your doctor's appointment. Okay. Raise your hand if you remember what the exact weather was like on the date of this doctor's appointment. Impressive. Raise your hand if you think you could remember an exact quote from a conversation you had with your doctor. Okay? And lastly, raise your hand if you feel pretty confident that you could tell a detailed story about the day that you went to the doctor. Okay. All right, great. Moving on to the right-hand side of the room. I would like you to recall the day that you had your first kiss. Raise your hand if you remember any of the meals that you ate that day. Okay. Raise your hand if you remember what the weather was like. Okay. Raise your hand if you could remember what you were wearing. And raise your hand if you feel pretty confident that you could tell a detailed story about the day you had your first kiss. Great, proud of you guys. So the left-hand side of the room didn't really impress us in their memory recall, while the right-hand side of the room had a markedly better performance for an event that occurred way earlier in their lives, maybe. Cognitive neuroscientists might say that the emotional nature of a kiss caused the right side participants to actually change their brain's connectivity, making emotional connections to seemingly normal information like a dentist appointment easier to recall. So if we measured the amount of neural firing in the right side group, represented by glowing orbs, we might see something like this. What you're seeing is connectivity across all lobes in the brain, representing neuronal connections for memories across an extensive network. This process of efficient encoding is comparable to the way your body changes when you work out. The left side of the room only works out their biceps. They go to the gym and they lift a weight on their left-hand side only. And I'm sure this muscle will get strong, and I'm really proud of them, but that does nothing for their overall health. If we measure the right-hand side of the room, they did kind of what would be a full body workout. They have increased connectivity because they targeted multiple brain areas like their amygdala, their thalamus, their frontal lobe, and their hippocampus, therefore getting a great workout. So how can we translate this to our study techniques? Although this is an exciting finding, we know it's not feasible for every memory to be as emotionally charged as your first kiss, but there are ways to make seemingly mundane or boring information easier to retrieve. Cognitive neuroscientists suggest that you generate and test, organize, elaborate on information that you later wish to remember to achieve the best results. When you generate your own questions from existing material, you're taking an active role in studying. When you organize information on your own terms, whether it be by color, category, or a song you made up, you're creating a meaningful framework for your own individual mind. When you elaborate on information, you create an association or image between two things. For example, picturing a cow chewing on grass to remember that Moscow is the capital of Russia. All of these techniques could strengthen your neuronal networks much more than repetition ever could. So to many of my peers' surprise, there are countless neuroscience labs on Boston College's campus that do research on this very topic. I've been able to volunteer my time as a research assistant in the Cognitive and Affective Neuroscience lab under principal investigator Elyse Mackenzie for 2 years now. And in this lab, we study 3 main questions. How do we remember emotional experiences? What phases of memory are affected by emotion? And how are the interactions between memory influenced by aging and by our own individual differences? So we're currently trying to answer these questions in a project called SEMO. That stands for something along the lines of social and emotional memory. SEMO investigates a curious phenomenon in cognitive aging. We're all aware that memory generally declines with age, but older adults show relatively preserved memory for information that is self-referential or emotionally arousing. So every Friday, I travel to the 5th floor of McGuinn and apply electrodes to a real live participant scalp, just like this. Through EEG data and various cognitive measures, we hope to discover some shared neural processes of self-referential and emotional information. If we can discover these mechanisms, even in older patients that resist memory loss, we would be able to leverage interventions that can prevent memory loss across the board. So now that we have all of this knowledge, let's take a step back. Where do we go from here? We can revisit the 3 points I made in the beginning of the presentation. Number 1, being all humans, especially those of us in academic environments, rely on and use our neurons to encode countless memories each day. Secondly, we can use neuroscience research to our advantage while we encode memories to enhance our academic performance. And last, we can take these techniques and use them to strategically combat cognitive aging. When I asked Professor Kensinger about the possible implications of SEMO's findings, I was really inspired by her optimism. She explained to me that the future of the aging process is becoming brighter as cognitive neuroscience research continues. We understand that many types of memory decline are pathological, not normal, and that the healthy aging process is possible. Most importantly, we now know that we have the ability to change our brain's neural networks into old age. What you do with this information is up to you, and if neuroscience research doesn't keep you up at night, I understand, but you should take some time to think about what does. If we can all ask questions that inspire us to find answers and drive change, I'm really excited about the direction that Boston College can go. Thank you.